Like any complex multi component system, the immune system is subject to failures of some or all of its parts. These failures can have dire consequences when the system loses its sense of self and begins to attack host cells and tissues. The result is autoimmunity. When the system errs by failing to protect the host from disease causing agents or from malignant cells, the result is immunodeficiency, the subject of this chapter. A condition resulting from a genetic or developmental defect in the immune system is called a primary immunodeficiency. A condition resulting from a genetic or developmental defect in the immune system is called a primary immunodeficiency. In such a condition, the defect is present at birth, although it may not manifest itself until later in life. Secondary immunodeficiency or acquired immunodeficiency is the loss of immune function. A condition resulting from a genetic or developmental defect in the immune system is called primary immunodeficiency. In such a condition, the defect is present at birth, although it may not manifest itself until later in life. So, secondary immunodeficiency or acquired immunodeficiency is the loss of immune function and results from exposure to various agents. By far, the most common secondary immunodeficiency is acquired immunodeficiency syndrome or AIDS, which results from infection with human immunodeficiency virus or HIV-1. In the year 2005, AIDS killed approximately 3.1 million people, including about 500,000 children under 5 years of age. HIV infection continues to spread to an estimated 14,000 people per day. That's scary. AIDS patients, like other individuals with severe immunodeficiency, are at risk of infection with so-called opportunistic agents. Opportunistic agents. These are microorganisms that healthy individuals can harbor with no ill consequence, but that cause disease in those with impaired immune function. The first part of this chapter describes the common primary immunodeficiency. It examines progress in identifying the genetic defects that underlie these disorders and considers approaches to their treatment, including innovative uses of gene therapy. Animal models of primary immunodeficiency are also described. The rest of this chapter describes acquired immunodeficiency with a strong focus on HIV infection and its effect on the immune system, AIDS, and the current status of therapeutic and preventative strategies for combating this fatal acquired immunodeficiency. Primary immunodeficiency. The primary immunodeficiency may affect either adaptive or innate immune functions. Deficiencies involving components of adaptive immunity, such as T or B cells, are thus differentiated from immunodeficiencies in which the non-specific mediators of innate immunity, such as phagocytes or complement, are impaired. Immunodeficiencies are conveniently categorized by the type or the developmental stage of the cells involved. Overall cellular development in the immune system, showing the locations of defects that give rise to primary immunodeficiency. The two main cell lineages important to immune function are lymphoid and myeloid. Most defects that lead to immunodeficiencies affect either one or the other. The lymphoid cell disorders may affect T cells, B cells, or combined immunodeficiencies, both B and T cells. The myeloid cell disorders affect phagocytic function. Most of the primary immunodeficiencies are inherited, and the precise molecular variations and the genetic defects that lead to many of these dysfunctions have been determined. In addition, there are immunodeficiencies that stem from developmental defects impairing proper function of an organ of the immune system. The consequences of primary immunodeficiency depend on the number and type of immune system components involved. Defects in components early in the hematopoietic developmental scheme affect the entire immune system. In this category is reticular dysgenesis, a stem cell defect that affects the maturation of all leukocytes. The resulting general failure of immunity leads to susceptibility to infection by a variety of microorganisms. Don't forget this name, reticular dysgenesis. Then, without aggressive treatment, The affected individual usually dies young in severe infection. Reticular dysgenesis. So, in the more restricted case of defective phagocytic function, the major consequence is susceptibility to bacterial infection. Defects in more highly differentiated compartments of the immune system have consequences that are more specific and usually less severe. For example, an individual with selective IgA deficiency may enjoy a full lifespan, troubled only by a greater than normal susceptibility to infections of the respiratory and genitourinary tracts. And lymphoid immunodeficiencies may involve B and T cells or both. The combined forms of lymphoid immunodeficiency affect both lineages and are generally lethal within the first few years of life. These arise from defects causing dysfunction in multiple cell types or in cellular interaction needed to develop immune response. They are less common and usually more severe than conditions resulting from defects in more highly differentiated lymphoid cells. B cell immunodeficiency disorders make up a diverse spectrum of diseases ranging from the complete absence of mature recirculating B cells, plasma cells, and immunoglobulin to the selective absence of only certain classes of immunoglobulins. Patients with these disorders usually are subject to recurrent bacterial infections but display normal immunity to most viral and fungal infections because the T cell branch of the immune system is largely unaffected. Most common in patients with humoral immunodeficiencies are infections by such encapsulated bacteria as streptococci and staphylococci and pneumococci because antibody is critical for the opsonization and clearance of these organisms. Because of the central role of T cells in the immune system, a T cell deficiency can affect both the humoral and the cell mediated response. The impact on the cell mediated system can be severe with a reduction in both delayed type hypersensitive responses and cell mediated cytotoxicity. Immunoglobulin deficiencies are associated primarily with recurrent infections by extracellular bacteria, but those affected have normal responses to intracellular bacteria as well as viral and fungal infections. By contrast, defects in the cell mediated system are associated with increased susceptibility to viral, protozoan, and fungal infections. So, intracellular pathogens such as Candida albicans, Pneumocystis carinii, and Mycobacteria are often implicated, reflecting the importance of T cells in eliminating intracellular pathogens. So, infections with disease viruses that are rarely pathogenic for the normal individuals, such as cytomegalovirus or even attenuated measles vaccines, may be life threatening for those with impaired cell mediated immunity. Defects that cause decreased T cell counts generally also affect the humoral system because of the requirement of Th cells and B cell activation. Generally, there is some decrease in antibody levels, particularly in the production of specific antibody after immunization. As one might expect, combined deficiencies of the humoral and cell mediated branches are the most serious of the immunodeficiency disorders. The onset of infections begins early in infancy, and the prognosis for those infants is early death unless therapeutic intervention constitutes their defective immune system. As described below, there are increasing numbers of options for the treatment of immunodeficiencies. The immunodeficiencies that affect lymphoid function have in common the inability to mount or sustain a complete immune response against specific antigens or agents. A variety of failures can lead to such immunodeficiency. Defective intracellular communication may be rooted in deleterious mutations of genes that encode cell surface receptors or signal transduction molecules. Defects in the mechanisms of gene rearrangement and other functions may prevent normal B or T cell responses. So, first is about SCID. SCID in the sense severe combined immunodeficiency. The family of disorders termed SCID stems from defects in the lymphoid development that affect T cells either alone or in combination with B cells and NK cells. Depending on the underlying genetic defect, a SCID patient may have one of several lymphoid cell phenotypes. All are characterized by defective T cell function, and dysfunction may also extend to B cells, NK cells, and both. because T cell defects are the common elements in SCID. A recently described screening test for SCID in neonates involves assaying excised DNA circles from the TCR locus in blood cells, which are evidence of TCR gene rearrangement. So, abnormal results from this assay allow early diagnosis prior to onset of life threatening infections. All forms of SCID have common features despite differences in their underlying genetic defects. Clinically, SCID is characterized by a very low number of circulating lymphocytes. There is a failure to mount immune response mediated by T cells. The thymus does not develop, and the few circulating C cells in the SCID patient do not respond to stimulation by mitogens, indicating that they cannot proliferate in response to antigens. Myeloid and erythroid red blood cell precursors cells appear normally in number and function, indicating that only lymphoid cells are depleted in SCID. So, SCID results in severe recurrent infections and is usually fatal in the early years of life. Although both the T and B cells may be affected, the initial manifestation of SCID in infants is almost always infection by agents such as fungi or virus that are normally dealt with by T cell immunity. Defects in the B cell lineage are not evident in the first few months of the affected infant's life because antibodies are passively obtained from a transplacental circulation or from mother's milk. So, SCID infants suffer from chronic diarrhea, pneumonia, and skin, mouth, and throat lesions, as well as a host of other opportunistic infections. The immune system is so compromised that even live attenuated vaccines, such as the Sabin polio vaccine, can cause infection and disease. The lifespan of a SCID patient can be prolonged by preventing contact with all potentially harmful microorganisms, for example, by confinement in a sterile atmosphere. However, extraordinary effort is required to prevent direct contact with other people and with unfiltered air. Any object, including food, that comes in contact with the sequestrated SCID patient must first be sterilized. Such isolation is feasible only as temporary measure pending treatment. A search for defects that underlie SCID has revealed several different causes for this general failure of immunity. A recent survey of 170 patients by Rebecca Buckley at, I'm not sure of the pronunciation, sorry for that, Duke University indicated that the most common cause, 78 case, was deficiency of the common gamma chain of IL-2 receptor, IL-2R gamma. Defects in this chain impede signaling not only through IL-2R but also through receptors for IL-4, 7, 9, and 15, because the chain is present in receptors for all of these cytokines. Deficiency in the kinase JAK3, which has a similar phenotype because the IL receptor signal through this protein molecule, accounted for 11 of these cases. A recent finding of an additional cytokine receptor defect found in 17 of the patients involved only the IL-7 receptor. These patients have impaired T but normal B cells and NK cells. Another common defect is the adenosine deaminase or ADA deficiency found in 28 patients in the study. Adenosine deaminase catalyzes conversion of adenosine to eosin, and its deficiency results in accumulation of adenosine, which interferes with purine metabolism and DNA synthesis. ADA synthesis results in defects in T, B, and NK cells. Defects in the recombinase activating genes RAG1 and RAG2 found in 5 patients impair normal TCR and immunoglobulin gene rearrangements and lead to an absence of functioning T and B cells but do not affect NK cells. Both immunoglobulin and T cell receptor genes require rearrangements to express the active forms of their products. The remaining cases included single instances or reticular dysgenesis and cartilage hair dysplasia, or were classified as autosomal recessive defects not related to known IL-2R gamma or JAK3 mutations. 2 of the patients had defects in the gene called Artemis, which encodes a DNA repair and recombination enzyme. In one case, a defect in the gene for the cell surface phosphate CD45 was found. Interestingly, this defect caused lack of alpha beta T cells but spread the gamma delta lineage and resulted in an elevated B cell numbers. 11 of the 170 cases were of unknown origin. with no apparent genetic defect or family history of immunodeficiency. Other known defects disrupt T cell function and give rise to SCID-like deficiencies. One defect is characterized by depletion of CD8 plus T cells and involves the tyrosine kinase SAP7 ZAP, an important element in T cell signal transduction. Infants with defects in ZAP70 may have normal levels of immunoglobulin and CD4 plus lymphocytes, but their CD4 plus T cells are non-functional. A deficiency in the enzyme purinucleotide phosphatase (PNP) causes immunodeficiency by a mechanism similar to the ADA defect. A defect leading to general failure of immunity similar to SCID is failure to transcribe the genes that encode class II MHC molecule. Without this molecule, the patient's lymphocytes cannot participate in cellular interactions with the T helper cells. This type of immunodeficiency is also called the Bayer lymphocyte syndrome. Molecular studies of a class II MHC deficiency revealed a defective interaction between a 5' promoter sequence of the gene for the class II MHC molecule and a DNA binding protein necessary for gene transcription. Other patients with SCID-like symptoms lack class I MHC molecule. This rare variant of immunodeficiency is ascribed to mutation in the TAP genes, which are vital to antigen processing by class I MHC molecule. This defect causes a deficit in CD8 plus mediated immunity characterized by susceptibility to viral infection.